we are now going to do this genetics activity that starts on page 170 of your book. And follow along as we go, filling in your worksheet. There will be some questions that this asks you that you'll go ahead and answer embedded in this video. And hopefully this will give a good overview of some basic genetics. So this activity describes using a gene for color. So notice that there's a difference between a gene, which is going to give instructions for making a protein, and everyone has a gene for this. So, for example, there's a gene for the amount of melanin that is produced in your skin, but different people have different versions of that gene, and those versions are called alleles. There's that bold word there. And the two alleles that we are going to focus on today are big A for normal enzymes and little a that's going to make a defective enzyme for color. So that defective enzyme is not going to be able to make melanin. So this goes through and reviews genotype and phenotype. And then it describes these kids and talks about their genotypes and their phenotypes on how to get both normal skin, and if you are not making proper amounts of melanin, then you are albino. So if you need review of genotype, phenotype, homozygous, heterozygous, dominant, and recessive, definitely read this paragraph, but I'm not going to read it to you. So our first question talks about the genotypes in our table, and it says, circle the genotypes in the table that are homozygous. So we have two different homozygous genotypes. Those are big A, and little a, and we end up with different phenotypes for those genotypes because little a's do not have any directions for making normal melanin in this example. So that's how we get albino. And big A only has directions for making a normal enzyme. Next question, what are two different genotypes for the albinism gene that result in the same phenotype? So look at your chart. Which two genotypes give you the same phenotype? So how do we get two people who have different genotypes ending up with the same phenotype? It's because in this situation, we've got big A and big A, both giving us normal skin and hair color. And that's because big A in this example is dominant, completely dominant to little a. So it doesn't matter what the second letter is. As long as you have one big A, you have enough enzyme to properly make melanin. So now we're going to go to the next section, and it's going to talk about how we inherit these characteristics from our parents. It reviews chromosome and meiosis and fertilization. And so we're actually going to do an example. I'm going to bring back our chromosomes from when we were doing crossing over. And in this part, it actually suggests that you're in a group, but we're not doing this in a group, of course. So we're going to use our model chromosomes to demonstrate meiosis and fertilization. And the pair of homologous chromosomes for each parent is going to include one chromosome with a big A allele. So this one has a big A, and one chromosome with a little a allele. And these are already duplicated because we're ready to start meiosis. So I'm just going to run through meiosis really fast. So we would get into prophase 1, where our tetrad would form. Then we would do metaphase 1, where all the chromosomes would be lined up in the middle. Then we would do anaphase 1 and telophase 1. and get something to mark so that we would end up with our 2 cells. Then each of these cells would go through this division again. We would be in prophase 2, metaphase 2, where things would be all lined up in the middle, anaphase 2, where we would split and split, and then we would end up with telophase 2, where we would end up each with our separate cell. So looking at one set of chromosomes, we would have little a's, and we would have big A's in half. So out of our 4, 2 of our cells have little a's, 2 of our cells have big A's. So that means that when we do meiosis, if you have a big letter and a little letter, half of the gametes would have a big letter and half would have a little letter. So here for mom, we can see if mom was big A, little a, then half of the eggs would have a big A and half of the eggs would have a little a. And I'll bet you could imagine if we did the same thing for dad chromosomes, half the sperm would have a big A and half the sperm would have a little a. Now, if this sperm meets this egg, what would we get as our zygote? Zygote would be the cell, that fertilized cell, that first diploid cell. Oops, I'm not even paying attention to what I'm writing. I'm talking and writing at the same time. So this one, big A and big A. So this zygote would be big A, big A. If this sperm meets this egg, then we would have a big A with a little a. Remember, in genetics, we write the capital letter first if there are 2 letters. Here, again, we would have this sperm meet this egg, and this sperm meet this egg. We would get this. So instead of drawing out all of our sperm and egg combinations, we generally just write this as a Punnett square. That is what a Punnett square is showing. If you've never thought about what exactly a Punnett square is showing, it is really showing what are the potential sperm and the potential eggs, and then what are the potential zygotes. So here we've got this Punnett square, and each parent is here on the outside, and all of these here are the zygotes. So, question 6 says the genotypes of the zygotes in the Punnett square represent the possible genotypes of the children of this couple. Explain why each child will have the same genotype as the zygote he developed from. So, if this zygote is big A, why will the kid, when he or she grows up, also be big A? And that's because once that zygote has formed, now the types of cell division is all mitosis. And mitosis gives identical DNA for every daughter cell. So the original cell is big A, so every daughter cell will be big A. Same thing for big A ones and little a ones. So, question 7, for a heterozygous mother, heterozygous is a big A, little a mother, what fraction of her eggs have a little a allele? That would be half. For a heterozygous father, what fraction of his sperm have a little a allele? That'll also be half. So, what fraction of this couple's children would you expect to be little a? The answer is a fourth. And there's a couple ways you could do that. You could think about this Punnett square. because that's what this is. And you could see, oh, 1 out of 4 is that little a couple, couple's child. Or you could use probability rules. Some of you all might like probability, and the probability of 2 little a's, if you know the probability of this little a is a half, and the probability of this little a is a half, the probability of getting them together, you would multiply those together. So 1/2 times 1/2 is 1/4. So if you want probability rules. So now we're going to try a few problems. So it says complete this Punnett square for 2 parents who are homozygous big A, big A. So that means each parent is big A, big A. So here's one parent. Here's the other parent. What would the kids be? They will all be big A, big A. How about this one? Now the parents are both little a, little a. So one parent on one side, one parent on the other. What would the kids be? All the kids would be little a, little a. Now this one says complete the Punnett square for a mother who's heterozygous big A, little a, and a father who's homozygous little a, little a. So mom goes on one side, a big A and a little a. Dad goes on the other side, a little a and a little a. Now we'll need to figure out the kids. Question 9 says for each of the Punnett squares, it says the 4 Punnett squares on this page, and that's because originally the spacing was different and it included this one over here. So I'll do this one as an example, and then we'll flip back here. It says for each of the 4 Punnett squares, indicate the phenotypes of each parent and each child. So we've got normal versus albino. So big A is normal. Big A is normal. Big A is normal. Little a is albino. So we're going to put an O there for 0 melanin or albino. So now you decide what you're going to see here. So hopefully you see that all of the kids here are normal. All of the kids here are albino. And this one was half and half. So these were normal, those were albino. So it says, explain why 2 albino parents won't have any children with normal skin and hair color, but 2 parents with normal skin and hair color could have an albino child. So 2 albino parents would be little a, and little a. That X means that they're going to be mated or crossed. So if this is the 2 albino parents, one albino parent with another albino parent, there's no way to get a big A in there. But what's the way to have 2 parents with normal skin and hair color having an albino child? Hopefully, you figured out that if one parent, oops, I just drew through my little a, that if one parent was big A, little a, and the other parent was big A, little a, you could end up with an albino child there from 2 parents who were both normal pigmented. Question 11 says albino children are rare in the general population. Based on this observation, what is the most common genotype for parents? The most common genotype for parents is probably big A, because we don't see many albino children in the general population. There probably aren't very many little A's out there. Although this is um, an interesting scenario because most albino children actually are a result of a spontaneous mutation where there was a mistake in the copying of the DNA for the sperm or the egg that formed, and there's actually a broken gene in there. So that's how they would get the albino gene. Um, and I also don't want you to think that any genetic condition is actually recessive. Recessive does not mean rare, and dominant does not mean common. We're going to get into that a little bit more when we talk about genetic disorders soon, but it all has to do with if it's going to be able to be covered up or not. So I'll leave that for genetic disorder day, but just know that just because it's rare doesn't mean that it's recessive. So that's not why it's rare that it's recessive. Um, Other examples, for example, dwarfism. I will go ahead and tell you that one now. Dwarfism is actually a dominant trait, at least the most common type of dwarfism, called achondroplasia, is actually the result of a dominant mutation. But most of us aren't dwarfs, so most of us are whatever the little letter would be. So, like little h, if we were doing h for height. So most of us are little h, and there are very few big H's out there for dwarfism. So, all right, now for this section, we're going to look at some coin tosses. In this, you're going to use your 2 coins that I gave you in class, and or any 2 coins that you've got, and we're going to have a dominant allele. Big A is going to be heads, and a recessive allele, little a, is going to be tails. So you're going to have to do both of the flips instead of following the directions of finding someone to be your partner. So you're just going to figure out both parents. So to do this, it says that we need to do this in sets of families. So you're going to take your coins and you're going to flip them. And hopefully you can see that I got 1 heads and 1 tails. So for my first family of 4 kids, move my thing a little bit, I got big A and little a. So now I'm going to flip my coins again. I got big A and little a again. I'm going to flip again. Oh, this time I got heads and heads. So I'm going to have big A and big A. Get this. And now I'm going to flip another time. Look at that. I got little a and little a. So I'm going to count up. How many did I have of each kind? I had 1 big A, big A. I had 2 big A, little A's. I had 1 little, little. Now we're going to do this again. We're going to start over with our families. And we're going to start our count from 0. So I'm going to flip. So that was heads and heads. I've got tails and tails. I got heads and heads. I got tails and tails. So this time for this family, I got 2 big A, big A's, no big A, little A's, 2 little a, little a's. So you continue doing this with your other 2 families, and then you're going to total them up. So how many big A's did you get? How many big A, little a's? How many little a, little a's? And figure out your percents. So hopefully you have gotten your percents, and our predicted percents would have been 25, 50, 25. But then let's look here. It says, notice that even if a family already has a little a child or a big A child, a later child may have the same genotype as an earlier child. This occurs because the genetic makeup of each child depends on which specific sperm fertilized which specific egg. And this is independent of what happened during any of the previous children's fertilizations. So these predictions are for each child, not necessarily for a family. So each child has those chances each time. So, looking at our question, 2 big A, little a parents have 2 children with the big A, little a genotype and 1 child with the little a genotype. If these parents have a 4th child, what is the probability that the 4th child will have the big A genotype? The answer is 25%, because the big A is a 25% chance. Doesn't matter that they've already gotten the other numbers. So number 2 says, for each family of 4 children produced by your coin toss matings, compare your results with the predictions from the Punnett square. It says, use a check mark to indicate any family that has exactly the numbers predicted by the Punnett square. So for mine, I actually had one that did. Maybe you did too. So sometimes you might get exactly the right number or the predicted number, but you don't have to as well. Because again, this is just probability. We're going to skip the class data part, since that'll be more complicated than it's worth. Try to collect everybody's data so that you all can do this one question. But generally what you'll see is the more people or the more data that you have, the closer the overall numbers are going to be to the predicted numbers. All right, so now, the last section of this is all about sex determination. Is it a boy or is it a girl? So we've got X's and Y's, X and Y chromosomes. Males have both an X and a Y chromosome. Females have 2 X chromosomes. And the whole reason that boys become boys is because there's a specific gene on the Y chromosome that activates early in development. If that gene is not present or doesn't activate properly, the default development is actually girl. So that it tells you a little bit about this gene and a little bit about it here. It actually offers for us to show meiosis again. I'm hoping that you all have gotten the idea of meiosis. So we can just skip right down here to number 2. It says, draw a Punnett square which shows the inheritance of the sex chromosomes. Use X to indicate an egg or sperm with an X chromosome and a Y to indicate a sperm with a Y chromosome. So in girls, they have X and X. So females are XX. So what's the only option for their eggs to have? X. So females would have X or X. Males would have an X and a Y. So it turns out that half of their sperm would end up with an X chromosome. So we draw that here, and half of them would have a Y chromosome. So we could fill this in. just like before. So, what percent of children would you expect to be male? Hopefully you can see that 50% of kids are expected to be male. So we can test this. For some reason, this worksheet goes through a very complicated scenario of trying to count boys versus girls. We took a tally in class about boys versus girls. So figure out what percent of males are the families, first your family, and then what percent of males are your classmates' families. And for the predicted percent for the Punnett square, we predicted it would be about 50% male. So fill in both your mother's children percent and the children of the mothers of all the students in your class. So we can fill in those two. So final questions. If a mother's first child is a son, is the next child necessarily a daughter? No, it's a 50-50 chance. How about if a mother's first child's a daughter, is the next child necessarily a son? No, again, a 50-50 chance. If a mother's first two children are the same sex, is the next child necessarily the opposite sex? Again, no. Each child has a 50-50 chance of being a boy or a girl. So, again, hopefully you're going to see that the number for all of the kids in all the students in our class, all the children of all of the mothers in our class, will probably be close to 50, even if your own family isn't at 50. But there might be differences because it is definitely a random chance that it might not be exactly right, because each of these is random. So hopefully you warmed up a little bit about Punnett squares and genetics, and we will be doing more Punnett squares next time.